Welcome back. I am the Body Whisperer, and I am glad you are here. Before we begin, let us settle in together. Find a comfortable position. Let your arms grow heavy, your legs, your shoulders. Take a slow breath in and let it go. One more. In and release. Good. Now, while you rest here doing nothing at all, something quiet is happening inside you. Your body is working. It always is. Every hour of every day, without ever asking for thanks. I want you to get to know it a little better, to listen to it, to feel it, to appreciate it. Because the more you understand the quiet world beneath your skin, the more at home you feel inside your own body. So, come and meet Insulin Ivan, one small part of you that keeps working through the night while you sleep. This is the story of 2 hours of blood sugar and insulin to fall asleep to. There is nothing you need to do now, nowhere to be, nothing to figure out. Just breathe, close your eyes if you have not already, and let us begin. Every time you eat a meal, something extraordinary happens inside your body, and almost nobody knows what it is. Not the calorie count, not the vitamins. Something far more interesting. A tiny molecular messenger gets dispatched from deep inside your pancreas, floats out into the bloodstream, and begins knocking on the doors of your cells, one by one, in an orderly, patient, unhurried round that has been running since long before medicine gave it a name. That messenger is Insulin Ivan, and this is his story. Now here is something worth clearing up right at the start. Most people, when they hear the word insulin, picture a syringe, a diagnosis, something that belongs to other people, to people who are ill. Ivan finds this mildly offensive. He has been doing his rounds in every human body, every healthy, ordinary, unremarkable human body, for as long as there have been human bodies. He is a biological fact. He is as ordinary as a heartbeat and as essential as breath. He just never gets the credit. So tonight, just this once, at the start of things, we are going to follow him. Here is the world we are entering. Picture the bloodstream as a vast, slow-moving river system. Faintly golden, lit from within. The current runs through every corridor and chamber of the body, from the great vessels near the heart, all the way out to the smallest capillaries, thin as a whisper, pressed close against muscle and fat and liver and bone. Everything the body needs to run, oxygen, fuel, signals, instructions, travels by this river. And floating along it right now, just after a meal has been digested, is glucose gloria. Gloria is the body's primary fuel. She is a simple 6-carbon ring of a molecule, golden yellow, cheerful, practically humming with potential energy, and she has just arrived in the bloodstream from the small intestine where the meal was broken down. She is ready. She wants nothing more than to get into cells, to be burned for energy, to be useful. That is her whole purpose. She just needs someone to open the door. That someone is Ivan. Ivan is compact, hexagonal. A small, faceted cluster of blue-gray that looks, if you squint, a little like a polished river stone with a face on it. He has 2 short, stubby arms and the general demeanor of someone who has been doing a very important job for a very long time and has learned not to rush it. He does not carry Gloria. He does not escort her to the door and push her through. That is not how this works. The rules, Ivan will tell you, are simple. The cell opens. Gloria enters on her own. What matters is the knock. Ivan is made in a very specific place. Deep inside the pancreas, that pale, elongated organ tucked behind the stomach, handling both digestion and blood sugar simultaneously. As overworked as any night shift manager you have ever met, there are tiny clusters of specialized cells called the islets of Langerhans. Inside those clusters are the beta cells, and the beta cells are Gloria's early warning system. When she rises in the blood after a meal, the beta cells detect the rise. Within minutes, they release Ivan into the bloodstream. Not one Ivan. Thousands. Tens of thousands. A whole dispatch of patient compact messengers sent out to every corner of the body. That is Pancreas Pete's operation. Pete is calm, dependable, never off the clock. He manages the Betafeld dispatch desk with the quiet competence of someone who has run this particular shift so many times he could do it in his sleep, which in a manner of speaking, he often does. He does not look up when Ivan ships out. He trusts the process. Ivan's destination is the plasma membrane, the outer wall of every cell in the body. And on that outer wall waiting is receptor Rita. Specifically, she is an insulin receptor, a Y-shaped molecule embedded in the cell's outer membrane, like a gatekeeper built into the door itself. Her 2 arms reach outward into the fluid surrounding the cell, scanning, listening. She is densest on liver cells and fat cells. Those are Ivan's busiest stops. But she is present everywhere. She knows Ivan's shape. She has known it for a long time. When he arrives and docks with her, something remarkable happens. She changes. She sends a signal inward, into the cell, a cascade of molecular instructions that says, in effect, glucose is here, open the door. In skeletal muscle cells, that signal ends with a protein called GLUT4. GLUT4 is a glucose transporter, a channel that normally waits inside the cell in a kind of storage position. When Rita receives Ivan's knock and fires her signal, GLUT4 moves. It travels to the membrane. It plants itself in the wall. And now Gloria, floating past in the bloodstream, has an open door. She goes in. She gets to work. This is Ivan's ordinary round. Knock, signal, door, glucose in. Quiet, orderly, continuous. What happens when the knock arrives and the door is slow to answer? What happens when Rita hears Ivan just fine, but the signal she sends inward grows faint, blunted, like a phone call losing signal one bar at a time? What happens when Gloria piles up in the bloodstream, waiting, and the cells are not letting her in the way they used to? And what does any of this have to do with the hours of sleep you are already beginning to drift toward? Ivan is stepping up to the first door of the night. He raises his arm. He knocks. We are just getting started. He knocks. We are just getting started. The first stop on Ivan's round is not the muscles. It is not the fat cells. It is a place that gets very little credit in polite conversation, a large rust-colored warehouse that runs the most complicated overnight accounting operation in the known universe. It is the liver. Liver Lisa, as she is known in these parts, is a broad, two-lobed organ sitting just beneath the ribs on the right side of the body. She receives everything the gut absorbs, every drop of digested meal, before it reaches the general circulation. She has been doing this her entire career. She does not take vacations. Her job, in plain terms, is this: when glucose is plentiful, she stores it. When glucose runs low, she makes more and sends it out. She is simultaneously the body's sugar warehouse and its sugar factory. The fact that she does both things and switches between them on a signal is one of the more remarkable feats of biological management you will ever encounter. And what is that signal? When Ivan arrives and binds to Lisa's receptors, and she has many of them, some of the densest insulin receptors in the body, she gets a very clear message: glucose is coming. Stop making it. Start storing it. Lisa, for her part, obliges. She converts glucose into a compact storage form called glycogen. Think of glycogen as glucose packed into a tidy bundle, the way you might compress a sweater into a travel tube before a flight. It takes up less space. It is ready to be unpacked when needed. Receptor Rita, the Y-shaped gatekeeper who lives in the membrane of every cell Ivan visits, handles the handshake on Lisa's behalf. Rita's two upward-reaching arms recognize Ivan the moment he drifts near. She binds him, and then quietly she gets to work. Here's what Rita does not do. She does not carry glucose herself. Neither does Ivan. This is the part that surprises people. Insulin does not transport glucose. Insulin does not barge through the door with a delivery cart. Ivan knocks. Rita answers. The cell then reorganizes itself to let glucose in. The cargo and the key are entirely different things. Ivan is the key. The cell unlocks its own door. In the liver, the mechanism is different from muscle, and this is worth slowing down for. Lisa does not rely on the GLUT4 transporter that Ivan summons in skeletal muscle. Her membranes use a different transporter entirely, one that is always present, always open, always letting glucose flow in passively, according to concentration. What Ivan's signal controls in the liver is not the door itself. It is what Lisa does with the glucose once it is inside. He tells her to store it, not release it. He turns off her glucose production machinery. That machinery is called gluconeogenesis, a word that simply means making new glucose. The liver runs this process continuously when the body is fasting, to keep blood glucose from falling too far. But after a meal, when Ivan arrives in force, gluconeogenesis is supposed to quiet down. Lisa gets the memo. She stops the factory. She switches to storage mode. This is a critical handoff, and it depends entirely on Lisa hearing Ivan's knock clearly. Meanwhile, across the bloodstream, Glucose Gloria is making her rounds. She is a bright 6-carbon ring of energy, warm as sunlight, and she has one purpose: to get inside cells and be useful. She moves through the blood cheerfully enough, but she cannot push open a single door by herself. She needs Ivan's signal to have arrived first. She needs Rita to have answered the knock. She needs the cell to have reorganized its interior to receive her. Gloria, if she is being honest, finds the waiting a little undignified. She is useful. She wants to be used. Every moment she spends circulating in the blood, rather than being taken up by a cell, is a moment she is not doing her job. She is fuel, sitting in the driveway instead of in the engine. But the system, when it is running well, does not make her wait long. Ivan binds Rita. Rita fires the signal inward. The cell opens. Gloria enters. She is converted to energy. The whole transaction takes moments. It is one of the quieter miracles of the post-meal body. Pancreas Pete, back at the dispatch center nestled behind the stomach, watches all of this through the measure of blood glucose itself. Pete is a pale, elongated organ with a broader head and a tapering tail, and he wears the expression of a man who has been managing 2 departments simultaneously for decades and has made peace with it. His exocrine side handles digestive enzymes. His endocrine side, specifically the beta cells tucked into small island clusters called the islets of Langerhans, handles insulin. When blood glucose rises after a meal, Pete's beta cells detect the change and begin releasing Ivan into the bloodstream. Not all at once, in a measured 2-phase process. A quick burst first, drawing on insulin already made and stored. Then a slower, sustained second wave, newly produced insulin, as the meal continues to be absorbed. Pete does not guess. He does not estimate. He reads the glucose concentration in the blood with the precision of an instrument panel, and he calibrates the release accordingly. Right now, the evening meal has been absorbed. Blood glucose has peaked and is beginning to slope gently downward as Ivan does his work. Pete is watching that slope. He is adjusting the release. Lisa is switching modes at the liver. Gloria is finding her way through open doors. Everything is proceeding as it should. For now. Everything is proceeding as it should. For now. Ivan moved on, and here's where the story gets genuinely interesting, because Ivan's next stop is not a single door, it is a whole district. Miles of it, if you measured it out. Skeletal muscle, the tissue that makes up the arms, the legs, the back, the shoulders, is the largest consumer of glucose in the human body after a meal. Not the heart, muscle. and Ivan has rounds to make across every inch of it. He had been here thousands of times before. The muscle cells, each one long and cylindrical, like a very patient tube waiting to be filled with purpose, had their own version of receptor Rita posted at every door. And Rita, as she would be the first to tell you, is not a passive fixture. She is the door. She does the deciding. Ivan knocks, and she determines what happens next. Now, here's the part that most people never hear about. And it is worth slowing down for because it is one of the more elegant pieces of biology this body has running at any given moment. Ivan does not carry glucose. He never has. This surprises people when they first learn it. They imagine insulin as some kind of delivery truck, pulling up with a cargo of sugar and offloading it at the cell. That is not even close to how it works. Ivan is a messenger, a signal, a knock on the door. The glucose, that is Gloria's job, and she is waiting in the bloodstream, doing her own part. Ivan just makes the door open. So how exactly does that work in muscle? When Ivan reaches a muscle cell and finds Rita on the outer membrane, he binds to her. That is the knock. Rita, a Y-shaped protein, sitting partly outside the cell and partly inside it, recognizes him immediately. She changes shape, just slightly, and that small physical shift sends a message inward. Not a word, not a signal you could see. A cascade of chemistry, one molecule activating the next, like dominoes falling through the interior of the cell. And at the far end of that cascade, something remarkable happens. Deep inside the muscle cell, there are these tiny transport proteins sitting in reserve, parked, waiting. They are called GLUT4, glucose transporter type 4, if you want the full name, though the name is less important than the image. Think of them as little revolving doors, folded up and stored away from the surface, doing nothing until the signal arrives. When Ivan knocks and Rita answers, those stored GLUT4 transporters get the message. They mobilize. They travel to the surface of the cell, to the outer membrane, and they embed themselves there, opening up. And the moment they open, glucose can move in. Gloria, who has been circulating in the bloodstream just outside, passes through those transporters and enters the cell. The door opened. The fuel arrived. The cell got what it needed. Ivan did not push. He did not carry anything. He simply knocked, and the whole sequence followed from there. Rita, if she were summarizing it, would put it plainly. Ivan provides the signal, she said. The cell does the rest. She was precise about this. She had no patience for the oversimplification that insulin moves sugar into cells. Insulin changes the cell's behavior so that the cell moves sugar into itself. That distinction matters, and it matters especially because of what can change. In a well-functioning system, the whole sequence is remarkably fast. Ivan binds, Rita shifts, the cascade fires, GLUT4 moves, Gloria enters. The timeline from knock to door open is measured in minutes. After a meal, muscle cells across the body are pulling in glucose at a healthy clip. and blood glucose levels return toward their usual range as a result. But muscle is also where a particular kind of trouble first becomes visible. The kind that can develop slowly and quietly over a long stretch of time, when the cellular response in muscle begins to be blunted. When the same knock from Ivan produces a smaller, slower GLUT4 response, the muscle takes up less glucose. Not none, less. The door still opens a little, just not all the way, and the glucose that does not make it into muscle has to go somewhere. It stays in the blood. Gloria, who just wants to be useful, finds herself circling. This is the biological definition of insulin resistance, at least in muscle. The impaired response of the tissue to insulin signal. Ivan's knock is intact. The message travels, but the door opens only part way and the cell takes in less than it should. It is not a broken hormone. Ivan is fine. He is doing his job exactly as he was built to do it. The problem lives downstream, in the cellular machinery that translates his signal into action. And here's the quiet part of all this, the part that is easy to miss if you are not paying attention. Muscle is the largest glucose-consuming tissue in the body after a meal. So when its response is blunted, the effect on overall blood glucose is not small. It is substantial. The arithmetic is simple. Less glucose into muscle means more glucose remaining in circulation. Gloria was starting to notice the math herself. She was circling a few more doors than usual, waiting a beat longer than she expected. Ivan, for his part, noted it, filed it away, continued his round. He had more doors to knock on, and somewhere back at Pancreas Pete's dispatch office, the numbers were quietly being watched. He had more doors to knock on, and somewhere back at Pancreas Pete's dispatch office, the numbers were quietly being watched. Ivan moved on. He had visited the liver. He had worked the muscle district. Now his rounds took him somewhere that does not always get its proper credit in this story. The adipose tissue, the body's fat cells. People hear the word fat and they stop listening. That is a mistake. These cells are not the villains of the piece. They are, in fact, one of the three most important addresses on Ivan's entire route: liver, skeletal muscle, adipose tissue. The three territories, the three tissues that between them determine how well the body handles glucose after a meal. Leave one out and you do not have the full map. Ivan knew this better than anyone. The fat cells, adipocytes if you want the formal name, though Ivan rarely stood on ceremony, are spread throughout the body, beneath the skin, around the organs. Tucked in places most people never think about until something else draws their attention there. And every single one of them has a door, a receptor, a place for Ivan to knock. The adipocytes are storage specialists. That is their primary job. When the body takes in more fuel than it can immediately burn, the fat cell is the long-term warehouse. It accepts the excess. It holds it. It does not complain about the hours. In a well-functioning system, insulin signal to a fat cell carries two messages at once. First, stop releasing the fat you have stored. Second, help take up the glucose that is circulating. Not every cell uses GLUT4 to do this. The precise machinery varies by tissue, but fat cells do. They share that transporter with skeletal muscle. Ivan knocks, the receptor responds, GLUT4 moves to the cell surface, and glucose enters. Neat, tidy, coordinated. Ivan arrived at the edge of the adipose neighborhood and straightened himself up. He was a compact thing, a hexagonal cluster of pale blue-gray that caught the light at certain angles and looked almost crystalline. Six-sided, faceted, friendly enough if you knew what you were looking at. He raised one of his small stubby arms and knocked. Receptor Rita answered. She was not the same Rita as the one back at the muscle cell. Each receptor is its own individual, occupying its own piece of membrane. But she might as well have been her twin. The same Y-shaped architecture, the same soft coral pink, the same upward-reaching arms that were really the extracellular binding domains, the parts of herself that reached out from the cell surface into the surrounding fluid, waiting for exactly the right signal. Ivan was the right signal. She recognized him immediately. Here's what the recognition looks like, and it is worth pausing on this for a moment, because it is quietly extraordinary. Ivan does not carry glucose. He has never carried glucose. He is not a delivery driver with a package tucked under his arm. He is more like a key, or rather not even a key, because he does not open anything himself. He is the signal that tells the cell a knock has been received. Rita does the rest. When Ivan binds to her, the receptor changes shape. That shape change is not a small thing. It sets off a chain of molecular events inside the cell, a cascade of signals passed from protein to protein like a message traveling down a long corridor, each messenger handing off to the next, the information moving inward deeper until it reaches the stores of GLUT4 transporters waiting in small internal pouches called vesicles. The vesicles move. They travel to the surface. They fuse with the membrane. The door opens, and Glucose Gloria, waiting patiently in the bloodstream just outside, can finally get in. Gloria was warm and golden and perpetually eager. She was a 6-carbon ring that simply wanted to be useful, and being useful meant being inside a cell, not floating around in the blood, waiting for something to happen. She had been waiting. She was good at waiting, but she was better at working. She entered the fat cell, and the fat cell did what fat cells do. It processed the glucose, stored what it needed, went about its business quietly and without fanfare, the way the body's best systems always do. Ivan gave a small, satisfied nod, insofar as a hexagonal peptide cluster can nod, and moved to the next door. The rules are simple, he had always said. Knock, wait for Rita. Let the cell do the rest. It worked the same in every tissue, liver, muscle, fat. The signal was the same signal. His shape had not changed since long before anyone associated his name with syringes and illness. He found that association faintly annoying, if he was being honest. He was not a medical emergency. He was the ordinary round. He was what happened after breakfast. Back at the dispatch office, Pancreas Pete glanced at his instruments. Blood glucose was moving in the right direction. The signal was reaching its destinations. The round was proceeding as expected. Pete was a pale, elongated shape with a broader head and a tapering tail, an arrangement that suited his dual responsibilities perfectly, since the broader end managed the blood sugar work while the tail handled digestion. He was calm. He was always calm. He simply watched the numbers, kept the beta cells at their stations, and waited to see if the response would hold. So far, it was holding. But the hours were long, and there were still questions the narrator had not yet asked out loud. Questions about what happens when the response does not hold, when the knocking meets silence, when the message arrives perfectly intact, and the door simply does not open the way it once did. Ivan moved on to his next address. He was not worried. He was never worried. He had rounds to finish. Ivan had made 3 stops: liver, muscle, fat. Each one a different conversation, each one the same essential request. Let the glucose in, stand down from production, keep things steady. He was methodical about it. He had been methodical about it for as long as anyone could remember. But here is something that tends to get left out of the story. Ivan does not work alone. Not because he needs help carrying things. He carries nothing, remember. The knock is the whole job. But because the body is not a simple system that only needs glucose levels to come down, sometimes they need to come back up. And when that moment arrives, Ivan's opposite number steps forward. His name is Glucagon Gerald. Gerald is a peptide hormone too, a messenger, like Ivan, born from the same neighborhood. The islets of Langerhans, tucked inside Pancreas Pete's long, pale body. But where Ivan is secreted by the beta cells, Gerald comes from the alpha cells, a slightly different department on the same floor. Think of it as the same building, different offices, very different job descriptions. Gerald's job is to raise blood glucose when it falls too low. He does this primarily by signaling the liver Lisa's department, to release stored glucose back into the bloodstream. The technical term is glycogenolysis, which is exactly as long a word as it sounds. The breaking down of glycogen, the stored form of glucose, back into its usable pieces. Gerald was not especially busy right now. Ivan's rounds had gone well enough. Blood glucose was settling. The post-meal rise was flattening into something calm and manageable, the way a wave smooths out after it breaks. Gerald was watching from a comfortable distance, arms folded, waiting to be needed. He considered this his natural state. Gerald had opinions about his role in the story of blood sugar regulation. He felt, and he would tell you this without much prompting, that he was chronically underappreciated. Everyone talked about insulin. Slogans, syringes, awareness campaigns. Gerald kept blood glucose from falling dangerously low between Ivan's rounds, between meals, through the long, quiet hours of sleep, and he rarely got so much as a footnote. But he noticed. The reason Gerald matters here in the middle of Ivan's otherwise orderly rounds is this: blood sugar regulation is not a one-direction system. Ivan pushes in one direction, bring glucose down, let cells absorb it, store the excess. Gerald pushes in the other, bring glucose up, release stores, keep the supply from dropping below what the brain needs. The brain, for what it is worth, is a demanding organ on this particular subject. It runs almost entirely on glucose. It does not store much of it, and it is not shy about making its requirements known when supplies dip. So the body keeps both hormones in rotation. Ivan active after a meal, quieting as glucose levels stabilize. Gerald stepping forward as glucose continues to ease down between meals or during the long fast of sleep. Back and forth, a seesaw that never quite touches the ground on either side. This is what physiologists sometimes call counter-regulation. One hormone regulates, the other counter-regulates. Together, they hold blood glucose within a range that keeps the brain supplied, keeps the cells fueled, and keeps things from going dramatically wrong in either direction. Ivan, for his part, acknowledged Gerald's contribution briefly. He was not given to lengthy acknowledgments. The rules are simple, he would say. You go up, I respond. You go too low, Gerald responds. It runs itself, which is roughly true. The system is largely automatic, operating below the level of any conscious instruction. Pete at the dispatch office monitors the glucose numbers and calibrates the output accordingly. More glucose coming in from a meal, more Ivan. Glucose stabilizing, Ivan production eases. Glucose dipping further, Gerald gets the call. It is elegant in the way that most biological systems are elegant. Not simple exactly, but balanced. A lot of moving parts that have learned over a very long time to move together. And here is the part worth sitting with for a moment. This two-hormone conversation is happening all the time. Not just after meals. Not just in the hours when a person is aware of what they ate. It runs through the quiet hours, through sleep, through the body's own slow rhythms. Ivan's rounds wind down as the post-meal glucose settles. Gerald picks up the thread. Between them, blood glucose stays within its working range without anyone having to think about it. This is the ordinary miracle of it. The body maintaining fuel availability for the brain and organs across a full day of meals, gaps, movement, and rest, using two small peptide hormones in a continuous, calibrated back and forth, is one of the more quietly astonishing things the human body does without being asked. Gerald would say that he deserves more credit for his half of that miracle. He is not wrong, but Ivan had already moved on. The conversation between hormones was ongoing. Ivan's personal round was not finished. There was still something to attend to. Something that happened not in the cells themselves, not in Pete's dispatch office, not in the careful counterpoise of Ivan and Gerald, but in what happened when the whole system ran long. When the knock went unanswered, not once, but gradually, persistently, over time. The mechanism had a name. It had 3 addresses: the liver, the muscle, the fat cell. Ivan already knew all 3 very well. He was about to explain what it looked like when the door was slow to open. Ivan paused at the edge of the bloodstream and considered how to explain this. He had been asked before. He was always being asked. People wanted something dramatic, a broken part, a villain, a switch that flipped. He never quite knew how to tell them that the truth was quieter than that. Here's what he usually said. The rules are simple. He knocks. The receptor recognizes him. The cell does the rest. That is the ordinary round. And it works beautifully, millions of times a day, in bodies that never think about it once. But sometimes, gradually, quietly, in a way that takes years to become visible, the response on the other side of the door begins to slow. Not the knock. The knock is fine. Ivan arrives the same way he always has. The same signal, the same shape, the same patient tap on the receptor. Receptor Rita recognizes him just as she always does. She binds him. She initiates the signal. Everything on the outside of the cell is working exactly as designed. What changes is what happens after. Inside the cell, the long chain of molecular instructions that Rita's binding is supposed to trigger, the cascade that ends with GLUT4 transporters moving to the surface, with the door opening wide for glucose Gloria, that chain becomes sluggish. Slower, less complete. Fewer GLUT4 transporters make it to the membrane. The door opens part way, or opens late, or opens for fewer molecules than before. Gloria is still out there. She has not gone anywhere. She is circulating, warm and willing, ready to be useful. But the door is not as wide as it was, and so she waits. While she waits, her concentration in the bloodstream stays higher than it would have been if the door had opened fully. This is insulin resistance. Not a broken messenger, not a lazy cell. The message arrives intact. The response is blunted. The signal travels in, and something downstream of Rita receives it less precisely than before. Ivan found it important to be clear about this. People often assumed something had gone wrong with him personally. He felt, somewhat too often cast as the problem. He was not the problem. He showed up. He knocked. The rules are simple. What changed was not his part of the exchange. Now here is the part that makes this whole system so quietly fascinating. This does not happen in every tissue at once, and it does not happen the same way in each. The three territories, skeletal muscle, liver, and adipose tissue, each have their own version of the story, and they are connected. What happens in one shapes what is asked of the others. In skeletal muscle, remember, the blunted signal means fewer GLUT4 transporters reach the membrane. That means less glucose enters the muscle cells after a meal. The muscle that would normally be the body's largest post-meal glucose consumer quietly steps back. It takes less, it needs less convincing to do so, unfortunately. So where does the glucose go instead? This is where Liver Lisa comes back into the picture. She was already managing her own post-meal workload. The signals telling her to slow glucose production, to shift into storage mode. But now she is receiving something else. The glucose that muscle did not take is circulating longer. Some of it will eventually find its way back to her. She takes it in, she processes it. She does not complain about this. She has been managing imbalanced loads since long before anyone named the condition. She once described her job in a way Ivan thought was very accurate. "We have order here," she said, "even when the order is inconvenient. Adipose tissue has its own variation. The fat cells, those quiet specialists along the body's storage depots, receive Ivan's signal through Rita just as the others do. But when their response is blunted, the effect is not only about glucose. Fat cells in a state of impaired insulin signaling also become less tidy in their management of the fats they carry. The details of that process are intricate enough to belong to a separate story. For now, the relevant point is this. All three tissues, muscle, liver, fat, are in conversation with each other. When one step lags, the others feel it. Receptor Rita, when asked to describe what a blunted response felt like from her side, was characteristically precise. She called it a calibration problem. The signal comes in clearly enough, she explained. It is what happens further along the chain. She was very insistent that this was not rudeness on her part. She initiates. She does her job. Something downstream receives the initiation with less precision than before. She found the distinction important and was not shy about making it. Ivan appreciated her specificity. He also appreciated that she never blamed him. That was, in a field full of misdirected blame, something he did not take for granted. The thing about insulin resistance that made it such a persistent puzzle for researchers and for bodies alike was not that it was catastrophic. It was that it was, for a very long time, invisible. The response is blunted, but not absent. The door opens, just not as wide. The glucose gets handled, just not as efficiently. And so the body keeps going. It keeps running. And something else, something quietly remarkable, begins to happen in response. Pancreas Pete had already noticed the numbers. He was watching them carefully, the way he always did, from the dispatch office in the center of the pancreas. The beta cells of the islets of Langerhans, those small industrious clusters that had been releasing insulin since long before anyone thought of naming insulin resistance, were beginning to calculate. If the first round of messengers was not quite enough, the answer was straightforward: send more. If the first round of messengers was not quite enough, the answer was straightforward: send more. And that is exactly what Pancreas Pete did. Pete, you will remember, runs the beta cell dispatch desk tucked inside the islets of Langerhans. Tiny clusters of specialized tissue scattered through the pancreas, like islands in a calm sea. His job is to read the numbers and respond. Blood glucose rises, he sends Ivan. Blood glucose settles, he eases off. It is an elegant system, simple on the surface, staggeringly precise underneath. But here is the thing about precision systems. They can compensate, and compensation, for a long time, looks exactly like everything working fine. When Pete's first wave of Ivans knocked on the muscle cell doors and the response came back a little quiet, fewer GLUT4 transporters reaching the surface, less Gloria getting in, glucose numbers sitting just a fraction higher than expected. He always notices. That is the whole point of him. So he made more insulin. Not dramatically, not in a panic. Pete does not panic. He simply looked at the dispatch board, saw that the numbers had not settled the way they usually did, and increased the production run. More compact hexagonal clusters of blue-gray clay, ready and waiting, released in careful pulses into the portal blood that flows straight from the pancreas into the liver. More Ivans heading out on the round. The technical name for this state, more insulin circulating in the blood than would otherwise be needed, is hyperinsulinemia. It is a word that sounds alarming if you meet it cold. It is not alarming. It is Pete doing his job. It is the system working exactly as designed, adapting in real time to a signal that is not landing with quite the force it used to. Think of it like this. Imagine you send a message to a friend and they do not reply. You send another, then another. You are not broken. The message is not broken. Something on the receiving end is a little slow today. So you try again, and eventually the message gets through. Pete is that kind of sender. Patient, methodical, unbothered by the extra effort. He has been doing this for a long time. And here's what makes this genuinely remarkable. The compensation works. For years, often. The beta cells ramp up production. More insulin circulates. More doors are knocked on with more frequency. And blood glucose stays in a range that looks, from the outside, completely ordinary. No signal fires. The system quietly absorbs the extra workload and keeps delivering results. This is why the whole process is so easy to miss. There is no moment where someone feels insulin resistance arrive. There is no morning where everything is suddenly different. Instead, Pete just gets a little busier. Ivan's rounds get a little longer. The dispatch desk hums at a higher volume, but it keeps humming. And from the outside, looking only at the final number, the blood glucose level, everything can appear exactly as it should. Glucose Gloria, for her part, appreciates the effort. She would far rather be let into cells, doing something useful, than circulating in the bloodstream, wondering when the doors will open. When Pete sends more Ivans, and more knocks land, and more GLUT4 eventually makes it to the surface, she gets where she needs to go. But she gets there. Liver Lisa, meanwhile, watches all of this with the quiet attention of someone who has managed imbalanced workloads before. When muscle takes up less glucose than expected, the excess has to go somewhere. Some of it comes to her. She processes it. She stores what she can. She does not complain about the extra load, but she notices it, and she files it away in the manner of someone who keeps very good records and does not throw anything out. Now, here's where the story gets genuinely interesting, in the way that biology often does when you follow a thread far enough. Researchers have been asking the question for some time, and it does not have a clean answer yet. The question is this: which comes first? Does the blunted cellular response cause the elevated insulin? Or does the elevated insulin itself contribute to the blunting? That is not a rhetorical question. It is an open one. There is serious scientific work suggesting that hyperinsulinemia is not merely a response to insulin resistance, that it may also be a driver of it, that the extra insulin circulating over time may itself contribute to the very sluggishness in the cellular response that caused Pete to send more messengers in the first place, a loop that feeds itself. Action producing consequence, producing more of the same action. Pete did not design this. No one designed this. It is what happens when a finely tuned feedback system meets conditions that run in one direction for long enough. The system adapts. The adaptation becomes the new normal. The new normal shifts the baseline. And the loop continues, quietly, without announcing itself. Ivan finds this philosophically interesting, in the dry way that he finds most things philosophically interesting. He is still doing his job. He is still knocking on doors. He is arriving in greater numbers now, which is not how he would have arranged things himself. But the rules are simple. When the round requires more messengers, Pete sends more messengers. And so, the dispatch desk keeps running. The beta cells keep producing. Gloria keeps moving toward whatever doors will open. What nobody tells you is how long this can all continue in the background, unremarked upon, before anything shifts enough to be measured from the outside. The answer, in most cases, is quite a long time. And so the round continues. Ivan knocking, Rita answering, Gloria moving through doorways, Pete at the dispatch desk, calibrating, adjusting, sending out more messengers when the numbers ask for them. Liver Lisa receiving whatever muscle could not take. The whole system running, quietly, continuously, in almost everyone, without a single conscious instruction from the person it belongs to. That is the part worth sitting with for a moment. You did not ask your beta cells to do any of this. You did not schedule Ivan's rounds. You did not remind receptor Rita to pay attention. Every piece of this machinery runs on its own, below the level of awareness, below the level of thought, in the deep biological background of a body that has been managing blood sugar since long before blood sugar had a name. Now, here is where the story takes a turn. Not a dramatic turn, not a cliff edge. More like a slow curve in a long road where, if you are paying attention, you notice the landscape has quietly changed around you. The road is still the road, but the light is different. The turn is sleep, and not sleep in the abstract, inspirational, get enough rest sense. Sleep in the very specific, biological, measurable sense. Sleep as a condition the body's glucose system actually responds to. Sleep as something that when it changes things Ivan can feel in his rounds. Here's what that means in practice. Researchers have run what are called glucose clamp studies. The name sounds elaborate, but the idea is straightforward. You infuse glucose into a person at a controlled rate, while also giving them a fixed, steady amount of insulin. Then you watch how much glucose you have to infuse to keep their blood sugar stable. If insulin is working well, if the cells are responding cleanly to the signal, you need to infuse quite a lot of glucose to compensate, because the cells are taking it up efficiently. If the cells are not responding as well, you need to infuse less, because less is disappearing. The rate of infusion then becomes a direct measure of how effectively insulin is doing its job. Researchers call it the glucose infusion rate. Ivan would simply call it his score for the evening. In one study, 9 healthy people, young adults, no underlying conditions, researchers compared 2 conditions. In one, participants slept for 8.5 hours. In the other, they were kept to 4 hours in bed. One night. That is all. One night of short sleep versus one night of adequate sleep. The glucose infusion rate dropped by roughly 25% after the short night. 25% in 9 people. Now, 9 people is a small number. The researchers said so. This is not a sweeping population finding. It is a careful, controlled observation in a small group, which is exactly how this kind of mechanistic research works. You hold everything else steady and change one variable, and you watch what happens to a specific number. What happened here was a 25% reduction in glucose infusion rate, reflecting less efficient insulin signaling in both the liver and peripheral tissues, meaning muscle and fat, after a single night of reduced sleep in otherwise healthy young adults. Ivan, for his part, found this professionally interesting. He had shown up. His signal was intact. He knocked at receptor Rita's door, just as he always does. But the response on the other side, the intracellular cascade, the GLUT4 mobilization, the whole downstream machinery that Rita sets in motion when she recognizes him, that response was measurably quieter after the short night. Not broken, not absent, quieter, less efficient. As if Rita had answered the door a little more slowly, opened it a little less fully. This is the part that tends to stop people for a moment when they hear it. Because we talk about sleep in terms of how it makes us feel. Rested or not rested. Sharp or foggy. We talk about mood, about concentration, about the particular quality of Tuesday morning when Sunday was short. We do not usually talk about sleep in terms of what it does to the conversation between Ivan and Rita, between a hormone and a receptor, between a knock and a response. But the body does not make that distinction. To the body, sleep is not a lifestyle choice or a productivity strategy. It is a biological condition with specific downstream effects, including effects on how efficiently cells respond to insulin signals. A glucose clamp study showed that clearly, even if only in 9 people, even if only after one night, and that 25% figure is worth holding gently, not with alarm, but with curiosity. Because what it points to is a mechanism, a real, measurable, physiological connection between the duration of sleep and the efficiency of insulin signaling. Not a suggestion, not a correlation someone noticed in a survey. A clamp study, a controlled measurement of a specific biological rate. Ivan does not find this alarming. He finds it clarifying. The rules, he would remind you, are simple. The signal has to arrive. Rita has to respond. And the conditions inside the body, including the conditions that sleep either maintains or disturbs, shape how well that response comes. Pete, at the dispatch desk, was already watching the numbers adjust. One night was enough for a detectable shift. He noted it without drama and sent more messengers when the response was quiet. That is what he does. But here, at roughly the middle of the night, the story opens up a second question. If one night produces a measurable shift in healthy young adults, what happens when it is not one night? The first study asked a single question. One night, 4 hours, 9 healthy people. And it found a measurable shift, roughly 25%, in the rate at which glucose could be cleared from the blood. That is not a small finding. One data point. A single knock on a door does not tell you what happens to the door over time. So the second study asked the longer question. 7 healthy adults. 4 nights in a row. 4.5 hours of sleep each night. Not 4 hours, 4.5. A seemingly small difference, and yet the study was not looking at the liver or at peripheral tissue in broad terms. It was looking somewhere specific. It was looking at the fat cells. And that brings us back to a character you met earlier, one who tends to get less attention than the liver and the muscle, but who is very much part of the same 3-territory circuit. The adipose tissue. Think of it this way. Ivan's round every single day covers 3 major stops: the liver, the skeletal muscle, and the fat cells. That is the trio. Liver Lisa manages the glucose supply from the inside, deciding when to store it and when to release it. The skeletal muscle takes up the lion's share of glucose after a meal. And the fat cells, the adipocytes, respond to Ivan's knock by opening their own doors, drawing in glucose for their own purposes, and also by listening to the signal that says, "Not right now, stop releasing stored fats." Receptor Rita sits on the surface of fat cells just as she does on muscle cells. Same receptor, same fundamental shape, same recognition of Ivan when he arrives. The signal that flows inward from that binding is not identical in every detail to what happens in muscle, but the principle is the same. Ivan knocks. Rita recognizes him. An intracellular cascade begins. And the fat cell responds. Now in insulin resistance, that response in the fat cell becomes blunted, just as it does in muscle and liver. The calibration drifts. Rita is still there. Ivan is still knocking. The signal is still real. But the downstream machinery moves more slowly or moves less completely, and the full response does not arrive. The second study measured exactly this. After 4 nights of 4.5 hours, researchers looked specifically at how well the fat cells, the adipocytes, were responding to insulin stimulation. What they found was a reduction of about 30% in insulin sensitivity in those fat cells. 30% in 4 nights in healthy adults. Pause on that for a moment. The study was small, 7 people. That matters, and it is worth holding clearly. Small studies do not write policy. They do not hand out diagnoses. What they do is point at a mechanism and say, "Look here. Something is happening here that is real enough to measure." and the mechanism they pointed at was not obscure. It was receptor Rita, doing her job on the surface of fat cells, and finding that after 4 consecutive short nights, her downstream response had become measurably quieter, not absent, not broken. Here is the thing about the fat cells specifically. They are not just passive storage, they are signaling tissue. When receptor Rita on a fat cell responds properly to Ivan's signal, one of the effects is the suppression of lipolysis. That is the process by which fat cells break down stored fat and release fatty acids into the blood. Ivan's presence, properly recognized, tells the fat cell, hold on, keep what you have stored. Do not release it yet. When that recognition becomes blunted, the suppression becomes incomplete. The fat cell becomes slightly more willing to release fatty acids even when Ivan is present. And those fatty acids circulating in the blood have their own downstream effects on how the liver and muscle handle glucose. It is, as Liver Lisa would say in her patient way, a cascade. One thing leads to another. The tissues do not operate in isolation. Pancreas Pete, at his dispatch desk, is keeping count through all of this. He does not know which study we are in. He only knows the numbers. And when those numbers begin to creep upward, when glucose is not being cleared as efficiently, his response is the one he always reaches for. He sends more messengers, more Ivan. But here is where the 2 studies, taken together, begin to suggest something worth sitting with quietly. The first study showed a blunted response in liver and peripheral tissue after 1 short night. The second study showed a blunted response in fat cells specifically after 4 short nights. Same direction, same mechanism. Different tissues, different durations. What they share is this: the effect showed up in healthy people with no prior diagnosis, no prior known metabolic difficulty. Receptor Rita was functioning just fine before the nights of short sleep, and then she was not functioning quite as well. Not because Ivan had changed, not because anything in the signaling molecule itself had shifted, but because something upstream, something about the conditions in which the cells were operating, had changed the cell's willingness or ability to respond. Glucon Gerald, Ivan's counterpart and self-described least credited colleague, was not involved in either study directly. But he would note, with professional composure, that when Ivan's signal becomes less effective, the balance of the whole 2-hormone conversation shifts. He is always listening. He is always ready. Ivan moves on. The night deepens. The cells are quiet, but not idle. And somewhere in the question of what 4 nights of short sleep can do to a fat cell's response, there's a deeper question waiting. One about what the body does to compensate when this happens not once, but consistently. The deeper question, as it turns out, is not a gentle one. It has edges. And the place to begin is with what the body does when it is a very good problem solver facing a problem that does not go away. Remember Pancreas Pete. Elongated, pale yellow composed, the dual role manager who runs digestion and blood sugar dispatch from the same office without ever getting a day off. When the first round of insulin went unanswered, Pete did not panic. He sent more messengers. That is the body's most reliable move. If the signal is not getting through, increase the signal. Blood glucose stayed in a reasonable range. Nobody on the outside would have known anything had shifted. That quiet compensation has a name: hyperinsulinemia. More insulin in the blood than the situation would have required if every door had opened promptly. Pete did not choose this because it was ideal. He chose it because it was the tool he had. And for a good long while, sometimes years, it is enough. Here is the thing about a compensation that works. It tends to run without drawing attention. The numbers stay acceptable. The system keeps moving. And somewhere in that steady, unremarkable functioning, a loop begins to form. Ivan explained it once to Glucose Gloria as they moved through the bloodstream together. She had noticed that there seemed to be more of him lately. More messengers on the same route. She asked him about it, the way she asks everything, directly, without ceremony, with that warm golden curiosity of hers. He paused. Then, in the way he has, short sentences, matter of fact, he said the doors are slower to answer. Pete sends more of us arrive, and the doors, after a while, may become slower still. Gloria thought about that for a moment. Then she said, "So the solution and the problem are the same thing." Ivan considered this. He said, "The rules are simple. The loop, less so." That loop is what researchers have been turning over for years, carefully, like something they found in the dark and cannot quite see the shape of. The question is this: when insulin resistance develops and Pete compensates with more insulin, does the higher insulin level itself make the resistance worse? Does chronic exposure to elevated insulin cause the receptors to become less responsive, a kind of overstimulation that eventually prompts them to dial back? Receptor Rita, for her part, has a phrase for it. She calls it a calibration problem. She is not wrong. When she is bound to Ivan again and again, with more frequency than the signal truly warrants, something shifts in how she handles downstream communication. Not a breakdown, a gradual recalibration. The door still opens, just a little more slowly than before. The science has not settled on which comes first. Whether the resistance drives the hyperinsulinemia or the hyperinsulinemia quietly deepens the resistance. What is clear is that the two states can reinforce each other. A loop, self-sustaining, running quietly in the background while the person goes about their day entirely unaware. And now, add sleep to that picture. 4 nights of 4.5 hours, 7 healthy adults. The fat cell's insulin response reduced by roughly 30%. That number from the previous chapter, small study, real measurement, describes something specific. It describes receptor Rita in adipose tissue receiving Ivan's knock and producing a noticeably quieter answer. Not no answer, a quieter one. The fat cell's job in this system, when insulin signals properly, includes suppressing the release of fatty acids into the blood. That suppression is part of what keeps the liver's workload manageable. When fat cells become less responsive to insulin, that suppression is blunted. Fatty acids move more freely. They arrive at the liver, in greater quantity. And liver Lisa, broad, chestnut brown, never off duty, processes what arrives. Lisa does not complain. That is not her style. She has Midwest directness and a talent for understatement. But she would note, if asked, that an increased fatty acid load is not a neutral event. The liver's handling of excess fatty acids can itself interfere with insulin signaling in the liver's own cells, which means that blunted signaling in fat tissue sends a ripple into hepatic tissue. The three-territory model, muscle, liver, adipose, turns out not to be three separate stories. It is one story with three addresses. So when sleep is reduced, even briefly, even in healthy young people who have no underlying condition, the fat cell response blunts. That blunting touches the liver. The liver's own signaling can shift. Pete's dispatch office picks up the signal and considers sending more messengers. More insulin in the blood, more frequent knocking. And the receptors, already recalibrating toward quiet, receive that elevated signal. The loop, in other words, does not require years to begin. It has a starting point. And that starting point can be as simple as a few nights of sleeping less than the body's machinery was designed for. This is not a verdict. It is a mechanism. The body is not broken. It is compensating, which is what bodies do. The compensation is a form of intelligence, a form of loyalty even. Pete keeps sending messengers because he believes in the round. Ivan keeps knocking because the rules are simple, even when the loop is not. Gloria floats alongside him in the bloodstream, still eager, still waiting for a door to open. She is not the problem. She never was. She is just waiting for the system around her to find its balance again. And that balance, it turns out, may have something to do with what happens in the hours before morning, while the body is doing something that looks, from the outside, like nothing at all. So here's the thing about sleep that nobody really explains. From the outside, it looks like nothing. The body is still. The breathing slows. The eyes are closed. Whatever happens in there, it seems quiet, passive even. A pause between the real events of the day. But Ivan has a very different view of that. Because while the waking world winds down, the body's internal economy does not clock out. It reorganizes, it recalibrates, and Ivan's role in that recalibration is far more specific than most people realize. Let's follow him into it. After a meal, Ivan's busiest hours are the ones most people are still awake for. The big post-meal wave, Pete dispatching messengers, Gloria moving towards cell doors, Rita signaling the GLUT4 cascade, Lisa standing down from glucose production. All of that happens in the first couple of hours after eating. That is the headline performance. But the night shift is quieter and in some ways more interesting. As sleep deepens, blood glucose is no longer rising from a meal. It is drifting gently downward or holding steady or ticking back up in small increments that Gerald manages from his corner. The counter-regulatory side of the conversation, raising glucose when it dips too far. Gerald, who considers himself chronically underappreciated, runs the overnight fast largely without fanfare. He signals the liver to release stored glucose in small, measured doses. just enough, not too much. The system is designed for this, and Ivan's workload is lighter at this hour. Fewer messengers in the blood. Pete's beta cells are not sending out the big post-meal wave. They are doing something more like maintenance, a low background hum of insulin secretion that keeps the system stable without swamping it. This low-level background is called basal insulin secretion. It is not nothing. It is continuous. It runs through every hour of sleep, every hour of waking, every stretch between meals. Ivan is never entirely absent from the bloodstream. The round never fully stops. It just changes character. During the overnight fast, basal insulin does one specific and important job. It tells the liver, Lisa, standing at her desk, managing glucose output, to keep that output measured, not too much. Not a flood. Just enough glucose into the blood to keep the brain and heart and everything else supplied, while the body waits for the next meal. Lisa, for her part, appreciates clear instructions. When Ivan's basal signal is clear, she runs an orderly operation. She releases glucose at a rate the rest of the body can use. No surplus piling up in the blood. No shortage leaving the brain scrambling. We have order here, she would say, and she means it as a statement of fact, not a boast. But here is the new wrinkle, and this is the moment worth sitting with. When the body has not had enough sleep, basal insulin secretion does not behave the same way. The signal Ivan carries is intact. Pete is still dispatching. But the tissues receiving that signal, Lisa's liver, the muscle, the fat cells, are less responsive than they would be after a full night of sleep. The glucose clamp study captured this in liver and peripheral tissue after a single night of shortened sleep. The fat cell study captured it specifically in adipose tissue after 4 nights. Both measurements were made in resting, fasting conditions, the equivalent of the quiet basal period, not the busy post-meal rush. Which means what the studies were actually measuring was this. What happens to Ivan's quiet background round, the maintenance shift, the steady hum, when the body has not had enough rest? And the answer was that the round becomes less effective, not broken, not stopped. Less effective. Rita is slower to answer the door. The downstream cascade produces fewer GLUT4 transporters at the muscle membrane. Lisa's responsiveness to Ivan's suppression signal, the signal that says, hold back, do not overproduce, is blunted. She produces more glucose than the situation calls for. Gerald's side of the conversation becomes a little louder than it should be, because Lisa is releasing glucose as if the counter-regulatory signal is stronger than Ivan's. The result is that blood glucose runs a little higher than it would after a full night of sleep. Not dramatically in healthy people with intact compensation. The kind of measurable that shows up in a carefully controlled study. The kind of measurable that Ivan, walking his quiet maintenance round, can feel in the silence between one knock and the next. There is a technical term for what happens to glucose levels in the fasting hours: hepatic glucose output. It is simply the amount of glucose the liver releases into the blood between meals and through the sleep period. Normally, insulin's basal signal keeps that output in check. In the studies of shortened sleep, that check was weaker. The output was higher. More glucose in the blood, not because more was eaten, but because Ivan's ability to tell Lisa to hold back was diminished. Lisa, to be fair, was not being unruly. She was reading a quieter signal and responding accordingly. She does not produce more glucose out of defiance. She produces more because the instruction was less clear. And Ivan, walking his quiet round in the small hours, knocking at doors that are slower to open, carrying a message that is landing with a little less force than usual, Ivan notices. He does not panic. He is not built for panic. He knocks again. He waits. He moves to the next door. The rules are simple. He just has to keep following them. Ivan has kept to his rules all through the night. Knock, wait, move on, knock again. The method has not changed since the first time Pete dispatched him. What changes, and what makes this biology so quietly astonishing, is the conversation happening between the territories he visits. Because here is something most people never consider. Liver Lisa, the skeletal muscle, and the fat cells, the three great insulin-sensitive territories, are not separate stops on an unconnected route. They are talking to each other, continuously. In a language made not of words, but of molecules. The primary dialect of that conversation is fat. Not dietary fat. Not the fat you might think of when you think of fat. The specific molecules at the center of this conversation are called free fatty acids. They are released from fat cells, adipocytes, through a process called lipolysis. That word, lipolysis, simply means the breaking down of stored fat into its components. One of those components is the free fatty acid. It gets released into the bloodstream, travels to the liver and to muscle, and delivers a message. Under ordinary conditions, Ivan handles this. One of the quieter things insulin does, less celebrated than the GLUT4 story, is suppress lipolysis. When Ivan knocks at fat cell doors after a meal, and Rita answers well, the fat cell does not just accept glucose. It also dials back its release of free fatty acids. The flood is reduced to a trickle. The liver gets a manageable signal. Muscle stays clear to do its own glucose work. Now consider what happens when Rita is slow to answer. When the fat cell's response to Ivan is blunted, as those 7 adults in the sleep study showed, with roughly 30% less insulin sensitivity after just 4.5 hours of sleep across 4 nights, the suppression of lipolysis is also blunted. The fat cell keeps releasing free fatty acids at a higher rate than it would if Rita were answering promptly. More free fatty acids in the bloodstream, traveling directly to the liver. Liver Lisa, who never asked to be the clearinghouse for everyone else's unfinished business, now has a heavier load arriving at her door. When the liver receives an elevated supply of free fatty acids, it does what any efficient processor does. It uses them. It redirects some into pathways that affect how much glucose it produces and releases. Here is the core of the message. High free fatty acid delivery to the liver is one of the recognized mechanisms that can impair the liver's own sensitivity to insulin. When Ivan knocks at Liver Lisa's door and tells her to hold the glucose, to stop releasing it into the bloodstream, a liver that has been steeped in a high free fatty acid environment is slower to comply. Hepatic glucose output stays elevated longer than it should. Ivan knocks. The message arrives. The response is muted. And so the liver releases more glucose into a bloodstream that already has more glucose than it needs, because muscle is also not pulling its share, because Rita on the muscle cell membranes is similarly working at reduced capacity. Glucose Gloria, for her part, finds herself standing in a corridor with doors on both sides and none of them opening at the usual speed. She does not panic. That is not her nature. She waits. She circulates. She trusts that the signal will eventually land. But the wait is longer than it used to be, and the corridor is more crowded than she remembers. This is the three-way conversation at its most concrete. Fat cell blunting drives higher free fatty acid release. Higher free fatty acids impair the liver's insulin response. The liver, impaired, continues producing glucose. Meanwhile, muscle, also less responsive, takes up less of what is already circulating. Pete, watching the numbers from the dispatch desk in the pancreas, sees the glucose not clearing as expected, and does the only thing available to him. He sends more Ivan, more messengers, more knocking, more attempts to open doors that have quietly become slower to respond. What is remarkable, genuinely remarkable, and worth sitting with as you drift, is that this entire relay is not a malfunction in any single part. Ivan is fine. Pete is doing his job. Gloria is doing hers. Lisa is processing what arrives. Rita is not broken. The machinery of each individual component is intact. What has shifted is the communication between them. The three territories have fallen slightly out of sync. The relay handoff, which under ideal conditions is smooth and continuous, has developed small gaps. And those small gaps, when sustained, require the whole system to work harder to maintain the same result. That is insulin resistance, seen not as a single event, but as a coordination problem across three interconnected territories. A relay team where the handoffs are just a little slower. The race is still being run. Nobody has dropped the baton, but the time on the clock is longer than it was. Ivan understands this. He has watched the relay long enough to recognize when the gaps are widening. He does not catastrophize. He adjusts his pace. He follows Pete's dispatches. He keeps knocking. The system is larger than any one messenger. And somewhere in that three-way conversation, fat cell to liver, liver to muscle, muscle back to the bloodstream, the body keeps negotiating a balance that most people will never consciously notice. Ivan finds something almost peaceful about that. The body working, even when the communication is imperfect. The negotiation continuing, even in the small hours. The negotiation continuing, even in the small hours. And now the night has gotten very deep. Not late in the way a clock decides, deep in the way the body decides. the way the whole system slows to something just above a whisper. Insulin Ivan is still here. He is always here. That is what most people do not realize about him. He does not clock out. He does not hang up his coat and call it done. His round gets quieter, the way a town gets quieter after midnight. But the streets are still open, and he is still walking them. His steps are smaller now. His knocks are softer. This is the basal round, the maintenance pass, not the urgent post-meal rush with glucose gloria flooding the bloodstream in a warm golden tide. Just the patient, methodical work of keeping the balance, the quiet version of what he does all day. Pancreas Pete is still at the dispatch desk. He has been there since before the sun went down, and he will still be there when it comes back up. That is just Pete, calm, dependable. Never entirely off the clock. The beta cells send out a steady, low murmur of messengers through the quiet hours. Not a flood, just a trickle, enough to keep the balance, enough to tell the liver to hold steady. Liver Lisa, for her part, is doing what she does. She always does. She receives the signal. She manages the stores. She keeps glucose output from climbing too high while the rest of the body rests. She would say she does not need to be thanked for it. She would probably say it, while already managing 3 other things. Receptor Rita waits on her membrane, ready. That is her nature. The attentive gatekeeper, always listening for the knock. She is the door. She does the opening. Ivan does not force anything. He never has. He arrives, he knocks, and Rita decides. In the deep quiet hours, she is simply waiting, poised, ready to respond the moment the signal comes. And glucose gloria, bright golden, eager glucose gloria, moves gently through the blood in the small hours, not the urgent wave of after a meal, just a quiet drift. Enough to keep the lights on. Enough to keep the brain fed, the heart steady, the slow machinery of rest running as it should. She is patient when she needs to be. She trusts the system. Glucagon Gerald is somewhere nearby, as always. The other side of the conversation, the counter signal, the one who steps in when glucose dips a little too low. He and Ivan have been doing this together for longer than either of them would bother to count. It is not a rivalry, it is a balance, a continuous, automatic, quietly astonishing balance. 2 hormones, 1 purpose. Keep the blood sugar where it needs to be, not too high, not too low. Through every meal and every fast, through every busy afternoon and every deep, still hour of sleep. This is the system, not a system for people with a diagnosis, not a syringe, not a medical word, not something that belongs to illness. This is the ordinary, unremarkable, extraordinary machinery that runs in every body, every single day. The one that starts before you are aware of it and keeps going long after you stop paying attention. Ivan's round has covered a great deal of ground: the liver, the muscle, the fat cells. The long relay of signals that crosses 3 territories and somehow, most of the time, lands exactly where it needs to. The knock, the recognition, the response, the glucose entering the cell, the balance holding. And when the response is a little slower, when Rita is a bit harder to rouse, when fewer doors open on the first knock, Pete sends more messengers. Quietly, without drama. The compensation running for years in the background, the numbers holding steady on the surface, the system doing what systems do, adapting, adjusting, continuing. This is not a story about things going wrong. This is a story about how hard the system works to keep things right. The body does not panic. The body does not catastrophize. It sends another messenger. It waits for the door. It keeps the balance for as long as it can, in the way that only something that has been doing this for a very long time can manage to do. Ivan is still walking his round. The night is still deep. The balance is still holding. The night is still deep. The balance is still holding. And somewhere in the quiet of it all, Insulin Ivan is still making his rounds. Not urgently, not frantically, just steadily, the way he always has. One knock at a time, one door at a time. The way he has done every night for as long as there has been a bloodstream to travel through. He is not in a hurry. He never was. That was always the thing about Ivan. He does not rush. He knocks. He waits. He trusts the process. The rules are simple, he would tell you, and they are. They have always been. Back at the dispatch desk, Pancreas Pete is keeping watch. His beta cells are doing what they always do in these deep, quiet hours, releasing a low, careful thread of insulin. Not a surge, just enough to keep the conversation going, just enough to let Ivan stay on the round. Pete does not sleep. Not really. He manages. That is what he does. Quietly, dependably, through every hour of the night. The dispatch desk hums. The numbers hold. And Liver Lisa, over in her corner of the body, is doing what she always does when the system is running well. She is keeping glucose production steady and low. Not too much. Not too little. Listening for Ivan's signal, responding to it, holding the line. We have order here, she would say. And she would mean it. She always means it. Rita is at her post. She is always at her post. Her two upward arms are ready, waiting for the next time Ivan comes to call. She is not asleep. She is not resting exactly. She is simply present, calibrated, quiet. When the knock comes, she will answer it. She always does. That is what she is there for. And glucose Gloria is drifting gently through the slow current of the bloodstream. There is no urgency now, no surge of glucose from a meal, no crowd of Glorias waiting at the door. Just a soft, even flow. The numbers easy, the current calm. She just wants to be useful. She always did. And right now, in the quiet of these deep hours, she is. Steadily, gently, doing exactly what she is supposed to do. Somewhere nearby, Glucagon Gerald is keeping his own watch. Not needed right now. The glucose is not dipping into territory that calls for him. But he is there. He is always there. Ready to step in if the numbers shift too far. Ivan's counterpart. The other half of the conversation that never actually ends. Gerald, for his part, is comfortable with the quiet. He does not need to be needed every moment. He just needs to know he is part of the story. And he is. The whole system is running. Every part of it. Not loudly, not dramatically. Just running. The way it does. The way it has always done. Through every dark hour, every slow breath, every moment of rest. This is the thing that is easy to forget. The body does not wait for you to pay attention to it. It just keeps going. Ivan keeps knocking. Pete keeps dispatching. Lisa keeps managing. Rita keeps listening. Gloria keeps drifting. Gerald keeps watch. All of it moving quietly in the background, while the world outside goes still, while the breathing slows, while the mind begins to let go. This is what the biology looks like from the inside, in the deep hours. Not dramatic, not alarming, just steady and careful and rather astonishing if you stop to think about it. Ivan is still on the round. The numbers are holding. The balance is still there. And there is nothing you need to do about any of it. It is not asking for your help. It is not waiting for your attention. It is simply doing what it does in the dark, in the quiet, in the slow and steady hours before morning finds its way back in. Let it run. Let Ivan knock. Let the doors open on their own. You have learned a lot these past 2 hours. The bloodstream, the signal, the knock, the transporter rising to meet it, the loop that compensates, the 3 tissues, the relay. The whole quiet conversation that runs beneath the surface of every ordinary day. And now, for a little while, there is nothing left to know. Only rest. Only the slow rhythm of a body that knows exactly what it is doing. Ivan is still out there, steady as ever, making his round. And here we are, the last quiet mile. Somewhere in the deep hours before morning, Insulin Ivan makes one more pass. Not a dramatic one. Not the urgent post-meal sweep with Gloria rushing through open doors and Pete calling numbers from the dispatch desk. This is the slow, steady basal round, the maintenance shift. The kind of work that never makes headlines because it never stops. The knock is soft. Rita answers at her own pace, warm and unhurried in the dark. The signal passes through. The cells hold their balance. Gloria drifts along the bloodstream at low tide, calm and content, not piling up anywhere, not waiting anxiously for a door that won't open, just being fuel in waiting. Lisa, over in the liver, holds her careful order through the long hours. She is not dramatic about it. She simply keeps the numbers from drifting too far in either direction, releasing just enough glucose to keep the brain and the heart fed while the rest of the body rests. "We have order here," she would say, if she were the kind to say it unprompted. She is not. She simply maintains. Pete sits at the dispatch desk, steady as a lighthouse keeper. The beta cells of the islets of Langerhans send their small, careful packages of insulin at a low, even rhythm. Not a flood, a drip, calibrated to a sleeping body's quiet needs. Pete has been doing this every night of your life. He does not need to be thanked. He does not need to be watched. He just needs the shift to keep going. And Gerald, patient Gerald, keeps his distance. Blood glucose is not falling. There is nothing for him to do. He waits in the amber quiet, ever professional, glad for once not to be needed. This is the whole system, at rest, doing its work. Not broken, not alarmed, not straining, just running, the way a well-kept clock runs. Not because anyone is watching, but because that is what it does. Ivan completes his round. The body is at peace. And now, the thing worth knowing is sleep takes you the rest of the way down. While you sleep, Insulin Ivan is already at work. Through the long stretch of the overnight fast, his basal signal flows at low tide, quiet, continuous, telling the liver to hold its glucose output steady, telling fat cells to stay settled, keeping the whole three-tissue circuit, liver, muscle, and adipose, in careful, unhurried balance. It is not dramatic work. It is the most important work there is. The kind of biological management that keeps your blood sugar from drifting too far in either direction, while your conscious mind is elsewhere entirely. He does it without your help. You don't have to do anything. Your body knows exactly what to do. It's been doing this work for your entire life. Every single night. Trust it. And as you drift deeper now, Insulin Ivan will be here when you wake up. quieter, calmer, better.